So we started off and we were very clear that viruses do not fit our definition of living organisms. And yet we're going to talk about how viruses evolve. While viruses can't create their own next generation and they can't propagate without their host, their genomes do change over time. And that really is the definition of evolution. It's a change in a gene frequency in a population over time. So viruses in general, their movement of evolution is from one that is highly infectious and harms the host to one that is still highly infectious but doesn't harm the host as much. And think about it this way. If you're a virus and you harm your host, you run the chance that you kill your host. And now your host is less likely to be able to pass on you, the virus, to other hosts. So what we generally see over time is that viruses evolve to be less of a killer strain and more of a spread everywhere strain. Now, that's a general rule of thumb. It's not always the case, but that's kind of the strategy that viruses take as they evolve. Now, another question you might have when you talk about virus evolution is where do they come from, right? So since viruses replicate only within cells, there's a chance that they could have actually come from cells themselves. So candidates for this source of viral genomes are plasmids. There's circular DNA in bacteria and yeast. Transposons. Transposons are bits of mobile DNA segments in our own genomes. So plasmids and transposons and viruses are all mobile genetic elements, and that's why they are possible candidates for the source of viruses and where they could have come from. So one example we might talk about is like a mimivirus. It's a double-stranded DNA virus. It's the largest virus discovered, and it's actually the size of a small bacterium. So it's like there's kind of controversy. Did they come from these plasmids and transposons? Are they relics? Are they relics of life before cells were here? And to be honest, we're really not quite sure. What we do know is that their genomes do evolve over time. So as we kind of focus on viruses, we do have to talk about their hosts. And we spend a lot of time studying them in animals, us. So some viruses cause infection to cells that produce toxins that lead to disease symptoms. And this is where we kind of figure out what's going on. Others have molecular components such as envelope proteins that are toxic to their host or whatever. But either way, viruses damage or kill cells when they're replicating themselves. And that causes disease in animals. And that's what we tend to spot. And so in an effort to kind of stop that, we create vaccines. Now, I often like to talk about the story of smallpox and cowpox. It was an early vaccine that was made, but smallpox was a virus that would ravage an area, and a lot of people would die from smallpox. But what they started to notice in some areas was that there was survivors, and that milkmaids tended to be on that list of survivors. And in one particular case, they noted that the milkmaids were milking cows that had cowpox. And so cowpox is not a zoonosis. It doesn't jump from cows to the milkmaid. However, the milkmaids got exposed to all this cowpox, which is a virus to cows, but not to them. And their bodies built up this actual defense to the cowpox that also worked for the smallpox. And so this kind of anecdotal example is kind of the idea of vaccines. They should be harmless derivatives of some kind of pathogenic microbe that stimulate the immune system to mount the defense. Now, when we say harmless, when they cause your body to mount an immune response, you might actually feel like you're sick. And so whether or not they're harmless is kind of up to debate. In general, there's a couple different categories of vaccines. And you guys will actually be doing a project on vaccines to explore them. But in general, we typically have where we have some other kind of vaccine or vaccine created from some other kind of pathogen that doesn't infect us. It's non-virulent, but it's close enough to the virulent type. That's kind of the example with smallpox and cowpox. Or we have some kind of attenuated vaccination where we've damaged the virus in somewhere where it shouldn't make you sick, but it's definitely going to mount an immune response. There are some other categories where instead of having a whole virus, we have parts of the virus that your body will respond to. Either way, you guys will explore vaccines in detail with that project, but vaccines can prevent certain viral illnesses, and viral infections can actually be treated with antibiotics. We do have some antiviral drugs, but for the most part, when you get a viral infection, it's got to run its course, and your body has to fight it off. So having a vaccination ahead of time can help your body fight it off, and more importantly, help you not become contagious to pass it on. There are a lot of people who will say that they actually feel better when they take antibiotics, even when they have a cold. What you've got to understand is that just because you have the cold virus, you've got an influenza virus causing a problem, that doesn't mean that you don't always also have some kind of bacterial infection, right? It's not always just the virus. And so if you took antibiotics and it made you feel better, it's likely that it was kind of curbing some bacterial infection. It had nothing to do with the virus. Your body has to beat the virus unless we have some kind of antiviral drug. Viral disease is a small, isolated and population can emerge and become global. We've seen this in a number of different cases. The most common and most relevant to us was COVID-19. So it was a new viral disease that emerged. This strain actually jumped species. In our case, we're pretty certain that COVID-19 came out of a laboratory where they were doing work. It was closely related to a disease that affects bats, and so it had some zoonosis properties, but it probably came out of a lab in Wuhan, China. So these strains cause pandemics or global epidemics. 2009, we saw a flu pandemic. It was swine flu. And, at the time we thought, oh my gosh, this is such a huge problem, my God. But 10 years later, we get hit with COVID-19 and things go worse, right? So this up here in the top left is H1N1, 2009. They're screening people and their temperatures and looking to try to see that they're wearing masks, trying to stop this pandemic. But COVID-19, this image right here is iconic for me because COVID-19, at least from my perspective and going through it, wasn't that scary until it hit Italy. And so this picture was March 21st, 2020 in Italy. And you can see these hallways lined up with patients suffering from COVID-19. COVID-19 led to widespread illness, significant loss of life, and it strained healthcare systems. And most people who lived through that remember everything just shutting down. It began in 2019 when we first identified the virus coming out of Wuhan, China, but it spread across the world. I do find it ironic that it was just about 100 years after the influenza Spanish flu in 1918. So it seems that our populations get exposed to these big pandemic things from time to time. But certainly COVID-19 will be something that for generations who went through it will never forget what happened. The last little bit we're going to talk about here is actually other infectious agents besides viruses. And these are virods and prions. Now, a virus has to have a genetic material and a protein coat. Well, these are like the two pieces of a virus. A viroid is a small circular RNA molecule that infects plants and disrupts their growth. So notice, it's just nucleic acid. A prions, these are slow-acting, usually hard to actually treat because they're slow-acting proteins that cause brain diseases because they propagate their misfolding. So prions are misfolded, but they convert normal proteins into the prion version. Scrapie in sheep, mad cow disease, and Creutzfeldt-Jacobs disease in humans are all caused by these prions. So if we look at that, what happens is the prions get into whatever location you've got that normal protein, and they spontaneously cause those proteins to misfold and they aggregate. And the crazy thing about prions is that we really just don't know a lot about how they infect their host, about how they work. We just know that they misfold these proteins. So these are two different examples besides viruses that infect populations.